Hello everyone, very good evening guys. Good evening students. How are you all? I welcome you all to this Biotechnika's YouTube channel. कैसे हैं सभी लोग? So before starting, please confirm if you can hear me and see me. Welcome Stunner. Hello, how are you? So you all may be knowing that today it is the 11th day of Nail the NEET revision series. So we have already completed our 10 lectures. So now it's 11 days and we are more with 4 days. So tomorrow we will be having a revision for chemistry or the coming days. So today we are going to deal with the biology section. That is genetics and inheritance. So today we are going to understand what is genetics, what are the principles of genetics, and what is the mechanism of inheritance behind this. So we'll understand this concept one by one. Firstly, we'll understand with and start with basics, and then we'll proceed to the principles of inheritance and the mechanisms behind it. And also, what are the mutational disorders that are leading to these variations. हेलो स्नेहा कैसे हो वेलकम एवरीवन वेलकम इनास so we'll wait for 1 or 2 minutes and then we'll start the lecture. So for your information, there is a 5 years previous year's NEET exam analysis. So if you want to know more about the NEET exam, what is the analysis of NEET exams and all about it, you can just find the link in the description below. You can just visit the link, find all the analysis that is there for the NEET exam. Welcome Inas. So I hope you can hear me and see me, right? So shall we start with the lecture? How's the Josh? Shall we start, all of you? So what is the topic for today's lecture? It is, sorry, wait, I'm not able to use this. Oh. So sorry guys, there was a technical glitch. So we are back now. So today I'm going to start with what is the principle of genetics and its inheritance. All right. So firstly, it is important to understand what is genetics. So what is genetics? Genetics is the study of principles of inheritance. Now, what is inheritance? Inheritance is the transfer of characters from parents to offspring. How the characters are transmitted from one generation to another. This is what called as inheritance. Now, what is variation? Once the character is being transmitted from one generation to another, it always leads to the variation in the characters. That means, what is the variation? What are the examples of variation? It can be the variation in the color of the skin, like dark skin, light skin, hair color, like it can be black or brown. So studying about the principles of inheritance and variation, this is what called as a field called genetics. So who is the father of genetics? Gregor John Mendel is the father of genetics. Why he is known as the father of genetics? Because he is the very first man who started his work on genetics. So firstly, he carried out hybridization experiment in garden pea. What is the botanical name for garden pea? It is Pisum sativum. So if you see the garden pea name, you can find a question from this in exam. So it is Pisum sativum. What is the span of work? He worked for this hybridization experiment for 7 years. And what is the year for this work? It is 1856 to 1863. Again, you may get a question from this year. What is the years or span of work in which Mendel worked? So it is always 1856 to 1863. And after that, this work that was done by Mendel, it was rediscovered by some other scientists like Bateson, Hugo de Vries, Teschermark, and here Bateson is known as the father of modern genetics. So who is the father of modern genetics? It is Bateson. But if the question is about the father of genetics, so it is Gregor John Mendel. So you can see here it is the image of Gregor John Mendel, who is the father of genetics but not modern genetics. And after this modern genetics, this genetics was proven by the experiments. And who was the first person to prove this experimental genetics? It was T.H. Morgan. So T.H. Morgan is known as the father of experimental genetics. So now we'll proceed further. So he selected pea plant for his study. Why he selected pea plant? What was the reason to select this pea plant? Because it was easily available and it also has a short lifespan. If the plant is having the short lifespan, it will be easily be completed in shorter period of time. So it has a short lifespan and also large progeny per cross. So every time when the cross is made between two pea plants, the progeny that is generated in the F1 F2 generation, it is always large progeny, matlab larger in size. More number of progeny are formed per cross. And also we can control crosses at every point of time. What is the meaning of control crosses? Control crosses, like if I want this is a bisexual flower. So if I want this flower to be only containing pistil, so I can easily remove the anthers from this flower. So it is called as emasculation. The removal of anthers is what called emasculation. So here controlling crosses were very easy. So now he selected pea plant. Firstly, he was working with 34 varieties of pea plant. What are the 34 varieties? And after this, he went down to 22. And finally, he worked with 14 varieties of pea plant. Here you can see 14 varieties of pea plant. And if you consider this as a contrasting character, so it is only 7. What are the 7 contrasting character on which Mendel worked? It is seed color or seed shape, pod color, pod shape, flower color, flower composition, as well as the stem height. So what are these contrasting character? So always the contrasting character can be dominant or recessive. So if we consider the height of the plant, the contrasting character is dominant, it is tall, which will always be expressing in the F1 generation. And the recessive character for this is short. What is the contrasting character for flower position? It is axial and terminal. What all characters you are seeing very first, it is always dominant. Dominant one, it will be expressed in the F1 generation. Now, what is the trick to recall this whole 7 trait? Because questions from this topic, it may ask in exam. So what is the trick for this? So the trick for this is Hi, Phi, Shilpa, Pandey. What is it? Height. H stands for height here. Height, it can be tall, it can be dwarf. But if it is tall, so it will be dominant character. The first one is always the dominant character that will be expressing in the filial first or F1 generation. F stands for flower. Now under flower, you may have the position of the flower or the color of the flower. If I'm considering the color of the flower, purple is the dominant character as well as white is the recessive character. Similarly for position, if the plant is bearing the leaves or flowers at the terminal position, that is what called as terminal flower position, or if it is bearing flowers at the axillary position, so it is called as axial flower position. Seed can be, we can classify this seed as shape or color. What is the shape of the seed? It can be round or wrinkled. What is the color? If the color of the seed is yellow, that means it is bearing the dominant character. If the color of the seed is green, that means it is bearing the recessive character. And also we can look at for the pot. If the pot color is green, it is the dominant character. If it is yellow, it is recessive. And what about the pot shape? If the pot shape is inflated, it is dominant. So if it is constricted or pinched, it is recessive characters. So he was working on 7 contrasting character. This contrasting, always we have 2 different kinds of characters for one trait. So 14 true breeding plant varieties. How 14? He selected 7 contrasting character, and for every character there are 2 traits, so we can just multiply it with 2, so 7 2 ja 14. So 14 true breeding pea plant varieties he was working on. So the first question that you have for this basics is, what were the years of the hybridization experiments that were carried out by Mendel? A, B, C or D? Here it is asking about the years of experiment that Mendel worked. Very good, Inas, Sneha. It is option D, because in the first slide only I mentioned the span of work. What is his span of work? It is 1856 to 1863. So the right answer for this question is option D. Now, in his classical experiments on pea plant, Mendel did not use. What he did not use for his experiments? Is it flower position? Is it seed color? It is pod length or seed shape? So here you have to recall the 7 contrasting characters that were used by Mendel. Stunner. Option C. Very good. Pod length he didn't use because he was using the flower position. How? One was terminal and one was axial. He also used seed color. If the seed color was yellow, it was having a dominant expression. If it is green, recessive. But he didn't talk about pod length. But here he talked about what is the pod shape. If it is inflated or constricted. And also he talked about pod color. What is the color? If the pod color is green, that means hara hara matar that we are taking from the market. So if hara color is there, green color is there, that means it is the dominant. अगर पीला होगा तो कोई लेना नहीं चाहेगा, ठीक है? So it should always be green in color. And also he talked about seed shape. So the right answer for this question is Mendel did not use pod length. So this is the right answer. So now coming to monohybrid cross. So when we are studying about the inheritance of a single character, that is what called as monohybrid cross. Mono stands for single. So what all characters did Mendel use? Out of 7 characters, Mendel used one of which is height of the plant. It can be tall, it can be dwarf too. So if it is a tall plant, so it will always have TT as a genotype. What is this T representing? T representing tall. And if it is a dominant allele, it will always have double capital T as a genotype. But if it is a dwarf, it will have a small double t. Now, what happens here? Meiosis happens, and meiosis will lead to the segregation of gamete, and out of which only one T is selected, and here also one T is selected. And when they are selved, they will form capital T and small t. This is what called as F1 generation. So if we look into the phenotype of a F1 generation, it is always tall. Why tall? Because it is bearing capital T and as well as small t. So whenever capital character or dominant character is there along with the recessive character, that is a small t, it always masks or hide the expression of recessive characters. So here in F1 generation, you will only see the expression of a dominant allele. And what is a dominant allele here? It is tall. So the plant height is tall. So here in F1 generation, you will find the plants that are tall in size. But after he carried out his experiment to see if the recessive character is lost in the F1 generation. So further, he served this plant TT with small tt. So what did he find? We'll make a Punnett square. So what did he find here? He found here in F2 generation, he was getting tall size plant. Again, TT, tall size, here also tall size plant, but somewhere he was getting dwarf size plant. That means in F2 generation, he was able to see the expression of dominant as well as recessive allele together. But in F1 generation, there was only the expression of dominant allele. That means the all the tall height of the plant, they were tall. But in F2, they were tall also, dwarf also. So how the genotypic and phenotypic ratio was calculated? So here if you see the phenotype, what is a phenotype? Phenotype is the physical appearance of a trait. So what is the physical appearance जो दिख सकता है, which we can see? That is what called as phenotype. तो दिखेगा क्या? Height of a plant, whether it is tall or it is dwarf. So here TT stands for tall. So here tall plant milega. Again capital T is there. So again tall, capital T is there, again tall, and here it is a small tt. That means the plant size would be dwarf. So what is the phenotypic character or ratio? It is 3:1. Means 3 times we are getting tall plant and 1 time only we are getting dwarf plant. Now what is the genotypic ratio? Genotypic ratio represents the genotype of an organism. What is a genotype? That is a genetic makeup. Now, what is a genetic makeup here? We are simply talking about capital T, capital T, capital T, small t, or small t, small t. This is what called the genotype. Now, what is a genotype? You can see one time we are getting capital T, capital T. That means 1. And twice we are getting capital T, small t, that is 1:2, and once we are getting again small t, small t. The genotypic ratio becomes 1:2:1. Now here how the questions are being asked for monohybrid cross. So generally they ask about the definition. If the inheritance, if we are studying the inheritance of only one character, that is what called as dash monohybrid cross. And also they ask about the phenotypic and genotypic ratio. But sometimes they are asking about what is the total number of phenotypes that are formed in a generation. So what is the formula to know the total number of phenotype? So if you want to calculate the total number of phenotype that is formed in the generations, that is 2 to the power n. And it is same for the gamete, again 2 to the power n. But for genotype, it is 3 to the power n. And for combinations, total combinations, if you need to, if the examiner is asking about total combinations, so be specific, there the formula becomes 4 to the power n. अब कैसे learn करना है? Total. We are talking about total things. So it is always higher. So 4. Phenotype and G. P stands for phenotype and G. So PG, you can call it as a code like PG. So PG, phenotype and gamete. जो छोटा spelling है, genotype is again G, but छोटा is gamete. So phenotype and gamete, उनका formula है 2 to the power n, whereas for genotype it is 3 to the power n, but for total combination it becomes 4 to the power n. Now what is n here? And here n is the number of heterozygotes. So now we'll take one numerical and see how it is happening. So let's consider we are having one hybrid AA, BB, CC, capital D, capital D. So if I'm asked to find out the total number of genotypes, so what formula will I use? So I will use the formula that is 3 to the power n. So n is the number of heterozygotes. So what is heterozygote here? Capital A, small a. Some hetero means different. So A, they are different. B, one is capital B, one is small b. One is capital C, one is small d. But in case of D, you can see this is homozygous. Both are similar. So we can find there are 3 heterozygotes here. So 3 to the power 3. So 3 into 3 into 3, that is 27. Okay? So here, sorry, this is 2 to the power n. For genotype, we are finding now, so it is 3 to the power n. But if the question is asking about phenotype or gamete, so it goes 2 to the power 3, or again for gamete, it goes 2 to the power e 3, that is 8. So I hope this is understandable to everyone. So sometimes they are also focusing on heterozygote. So what is the formula for a heterozygote? It is 2pq. Now what is 2pq here? P stands for dominant allele. Q stands for recessive allele. So there in the question, you will be given about the frequency of a dominant allele and frequency of a recessive allele. What you all have to do is, you have to just put a formula of 2pq. So if in the question it is given about dominant allele or recessive, just multiply it this. 2 से multiply करना है dominant and recessive को, then you will be able to find the frequency of a heterozygotes in a population. So after performing this monohybrid cross, Mendel proposed 2 laws of inheritance. The first is called as a law of dominance, and the second is called as the law of segregation. So the first law, that is law of dominance, Mendel told that like characters that we are seeing. What are characters? Characters are phenotypes. So all the phenotypes that we are seeing, it controlled by discrete unit that are called as factors. तो मेंडल ने बोला जितना भी phenotype we are seeing, that is controlled by factors. And what is factor here? It is nothing but genotype of an organism. सही है? Because in F1 generation we were seeing TT, and it is because of capital T we were seeing all tall plant. तो मेंडल ने बोला the character that we are seeing in the F1 generation, it is all because of these discrete unit that are called as factors. And what is a factor here? It is genotype. But Mendel को confusion हुआ. Mendel told that after this कि it is not like that कि recessive character it is lost in the F1 generation, but it may be there, it is masked or hidden by this dominant allele. Then he further proceeded his experiment and served this F1 progeny to get the F2 population. And then what did he finally find in the F2 population? He found that he was getting a tall variety of plant as well as the dwarf variety. And then he concluded that in F1 generation, we are only having the expression of a dominant allele, whereas in F2 generation, we are getting both the dominant as well as the recessive allele. And after that, he gave the second law of that is called as a law of segregation. This law is also called as purity of gamete, where Mendel explained that when gametes are forming in the, this undergoes meiosis and it will form gamete. How it will form gamete? Suppose this is capital T, this is small t, it will undergo meiosis and only one will be formed. These gametes are separated, these factors are separated in such a way that only one out of the 2 allele is selected. So Mendel told that in law of segregation that during gamete formation, this factor of the character that is present as a parent, they are segregated in such a way that only one out of the 2 allele is selected, and they are appeared in the F1 generation. So this is what called as law of segregation, and this is also known as purity of gametes. So now coming to crosses, what are the different types of crosses? So now if I consider in F1 generation, I am getting capital T and small t. So we have 3 type of crosses. One is back cross, one is test cross, and one is out cross. So from where we are starting in Mendel's experiment, we are starting with capital T as one parent population and small t as another parent population. And finally, the gamete formation is taking place, and what we are getting in F1 generation, we are getting capital T and small t. So if we are selfing this F1 generation with any of the 2 parents, so this is what called as back cross. Back cross means if we are selfing this F1 generation or this progeny that is formed in a filial first generation with any of the parent, if it is recessive or dominant or parent 1 or parent 2, then it is called as back cross. What about test cross? So Mendel told that if we are selfing this F1 generation progeny with the parent 2 or the recessive characters bearing plant, so it is called as test cross. That means selfing of an F1 with recessive characters. So what is the recessive character here? Small t, small t. And similarly, if we do it for a dominant character, then this is called as a outcross. So if capital T and small t, it is served along with the dominant character, that is parent 1 capital T, that is what called as outcross. So back cross, if we are selfing F1 progeny with any of the parent, whether it is parent 1 or parent 2, dominant or recessive, it is called as back cross. Test cross stands for if we are selfing this F1 progeny with recessive parent, and out cross when we are selfing this filial first generation with the dominating character parent. That is what called as out cross. So at a particular locus, frequency of allele A is 0.6. So the third question, the frequency of allele A is 0.6 and that of allele B is 0.4. What would be the frequency of a heterozygotes in a random mating population at equilibrium? So here you need to find the frequency of heterozygotes, and I've already explained about how to find the heterozygotes ka probability or frequency. There is one formula, so just you have to put that formula here. So what is the answer? D. Okay, anyone else? Okay, so the right answer for this question is, right Stana, you're correct. Right answer is 0.48. How we need to find this? 2pq. So what is 2pq here? 2 into p for dominant allele, that is 0.6, and the other one is given, that is 0.4. We just need to multiply all these values and it will give 0.48. This is the right answer. So the next question you have here is, to determine the genotype of a tall plant of a F2 generation, Mendel crossed this plant with a dwarf plant. This cross represents A. See how you have to solve the question. You need not to read whole question. Just find the catchy word, what it is saying. So it is crossing the tall plant of a F1 generation with a, or F2 generation with a dwarf plant. So the dwarf plant, we know it is a recessive character. So what is the answer? Answer D. What is the answer for this question? Answer B. Okay, so it is back cross. But I told, if we are crossing the F1 progeny with any of the recessive character plant or trait, then it is called as test cross. But if it is a dominating character plant or tall plant, then it is called as back cross outcross. But if we are crossing this F1 generation with any of the two parents, whether it is dominant or recessive, so it is called as back cross, because we are going back and crossing with the parental population. So the right answer for this question is, since it has a catchy word called a dwarf plant here represents recessive characters, or recessive means test cross. So the right answer for this question is option A. So how many gametes are formed from the mother cell having AA, BB, CC, DD genotypes? So again, you need to find about the gametes. How many gametes? That means total number of gametes. So what is the right answer for this question? How many gametes are formed for this genotype? AA, BB, CC, DD. Option C. Let's see how it is option C. AA, this is one of the heterozygotes. Again, one of the heterozygote. We have three heterozygotes in total. AA, BB, and DD. What about CC? It is homozygote. So what is the formula for finding the number of gametes? It is 2 to the power n. What is n here? N is the number of heterozygotes. So 2 to the power 3. Why 3? Because we have 3 heterozygotes here. So 2 to the power 3 is ace. So what is the final answer? Answer C is the final answer. So yes. Now coming to dihybrid cross. So if we are studying the inheritance of two characters together, that is what called as dihybrid cross. In monohybrid cross, we were studying the inheritance of one characters, but when we are considering or studying the inheritance of two characters together, it is known as dihybrid cross. So how we are studying this? 2:4:2, 1:2:1. So how we are studying the dihybrid cross? Suppose in previous slide we have seen it for the monohybrid cross, that is tall size plant and dwarf size plant. And when we are combining this character with one more character, suppose if I'm taking the seed ka shape if it is round or wrinkled. So if I'm crossing these two characters together, this is called a dihybrid cross. Why di? Because we are studying two characters together. And how the gametes will be formed now? Capital T, capital R, small t, small r. And further we will lead to the phenotypic ratio of 9:3:3:1, and here the genotypic ratio is 1:2:1, 2:4:2, and again 1:2:1. So how you have to remember this ratio for genotypic? Like in genotypic, we have seen in the monohybrid cross, it is 1:2:1. So just double it. 2:4:2, and again write 1:2:1. So this is the final genotypic ratio for a dihybrid cross. And what is phenotypic ratio? It is 9:3:3:1. So what is 9 here? 9 here represents we are getting the F2 progeny 9 times. कौन सा F2 progeny? पूरा dominant character. That means tall and round. What is 3 representing here? We are getting the recessive character of tall plant. That means dwarf we are getting, but the dominant character of a round seed. What is again 3 representing here? The dominant character of a tall plant, but a recessive character of a seed shape. That is wrinkled. And what 1 represents here? 1 represents here about the recessive characters of both the dwarf as well as the wrinkled seeds. So after performing this dihybrid cross, Mendel postulated third law of dominance, that is what called as law of independent assortment. So after studying this dihybrid cross, Mendel told that the inheritance of one character is independent of the inheritance of two characters. So how tall plant is delivering its character to next generation. It is totally independent of the round seeds or wrinkled seeds. So the inheritance of one character, it is independent about the inheritance of another character. This is what called as law of independent assortment. That means the genes are assorted independently. They are not dependent on each other. They're assorted independently. That is what called as law of independent assortment. And after coming to this, there were many exceptions to the law of dominance. So the Mendel's work that was criticized, and he had the exceptions for this. This is also called as non-Mendelian inheritance. What is this called? Non-Mendelian inheritance. So here we have three laws under non-Mendelian inheritance. The first law is incomplete dominance. What does incomplete dominance state? So incomplete dominance, it states that incomplete dominance. So previously in the Mendel's law, we have already seen that TT, when it is combined with a small t, we are getting T and T. One capital T, one small t. But the phenotype that we were getting is tall. But here it proved wrong. All time we are not getting the same phenotype. Not always tall characters we are getting in F1 progeny. This was proved by incomplete dominance. So what is this incomplete dominance? The genes or the part of allele that we are getting in first generation is incompletely dominant. That is not at all completely dominant. That means we can get the mixture of both the properties or both the varieties. So if it is T and small t, so we'll get an intermediate height plant, not tall always. So this is what called as law of incomplete dominance. So this was firstly studied in dog flower, that is known as strapdragon or Anterhinum majus. So how it was studied, we'll look into it because this is important in terms of exam point of view. So firstly, Mendel took the red color flower that is represented as double R, capital double R. This is the dominant characteristic, the flowers that are red in color. And he crossed with the small r, smaller, that is white in color. So during the gamete formation, it will have capital R and it will have smaller. So when these both traits were selved, it formed capital R and smaller. This time, the capital R does not code for the red color flower. But what did he found surprisingly? He found the flower was pink in color. Why pink? Because when you mix the red color with the white color, it will always give a lighter shade of it. And what is a lighter shade? That is pink. So here he was getting the pink flower. So further he said this pink flower. So how he said? What is the Punnett square for this? So if we write like this and make a Punnett square, so what will we get? Capital R, capital R, capital R, small r, capital R, smaller, smaller, smaller. So if we see the genotype, what is the genotype? Capital R, 1. Capital R, smaller, we are getting it twice, that is 2. And again, smaller, smaller, 1. And what is the phenotypic ratio? Appearance. One plant, it was appearing red, that is 1. Capital R, small r, that is pink. Capital R, small r, it is giving pink color flower. So again 2 and again smaller, that is a white color flower, again 1. So here you can see the genotypic and phenotypic ratio are same. That is why this is the exception to the law of dominance. तो law of dominance में क्या कहा गया था? Always dominant is expressed, but it is not like that. In case of incomplete dominance, both the varieties mixed together and they're expressed together in F1 generation. Like here we can see the pink color flower. This is what called as law of incomplete dominance, where you can have the equal ratio of genotype as well as the phenotype. And what is this ratio? 1:2:1, and again for phenotypic ratio it is 1:2:1. So again he studied this in starch grain size. So what is the reason for this starch grain size? So suppose there is a starch branching enzyme, that is what called as SBZ. So here if it is encoding BB character or small b, So when this genotype is capital T, capital B, capital B, it is always forming round or large starch granule. But when it was small b, it was always forming wrinkled small starch granule. But when Mendel combined both these properties, he found that capital B and small b that was having the intermediate variety. That means some seeds were small in size and some seeds were large in size. That is what called as incomplete dominance. Then what is codominance? When two characters that are expressed together, that is what called as codominance. So how we can study codominance here? Like it was previously studied in sickle cell anemia, and the second example is roan color in cattle. So now we'll see how. Suppose if I'm making this cow, suppose this is cow. This is brown color cow. And if we are selfing this with white color cow, so what will have? These both genes are expressed together. How they are expressed together? Suppose this is capital B genotype. This is small b, small b. So how the gametes will be formed? Capital B and obviously here small b. And when they are mixed together, it will give capital B, small b. So the cattle that we were getting here, it was brown color. But here it has a white patches on it. So when two alleles, when they are expressed together, so how they're expressing together? The cattle is brown in color, but it is having a white patches on its body. That is what called as a law of codominance. And what is multiple allelism? When many genes work together. So here we consider this in the case of ABO blood group. So here how the questions are being asked for multiple allelism. In general, the match of the following form. They'll give you the multiple allelism, and here you have to pair whether this is the right answer for this. And in front of this, they will write ABO blood grouping, and you have to match if it is a correct pair or not. So this is the correct pair. The example of multiple allelism is ABO blood grouping. So now, a pink snapdragon is crossed to a white snapdragon. What is the probability of getting a red snapdragon? Next question for you. What is the probability of getting a red snapdragon when you cross a pink snapdragon or a white snapdragon? So जब भी snapdragon का name आता है, so it is always signifying law of incomplete dominance, because this law, it was studied very fast in snapdragon, that is dog flower. So what is the answer? Option C, 1/4. So let us see what is the answer. So if I'm saying a pink flower, so what we were getting in the F1 generation, how the flowers were pink, it was having R and R genotype. So here you can see that it was having RR genotype, because red flower, it was mixed with white flower. So RR, it is mixed with the white snapdragon. What is the genotype for white snapdragon? It is small r, small r. So when it is mixed, how the gametes will be formed? R, and from here R will be selected. And what is the probability of getting the red snapdragon? Red here signifying about capital R, capital R. So what is the probability of this capital R, capital R? So when we solve this capital small r, what will get? So let's draw a Punnett square for this. So we'll get capital R, capital R, capital R, small r, capital R, small r, small r, small r. So what is the probability of red flower? So the red flower will have the RR genotype. So what is the probability? It is 1/4. So what is the right answer for this? Option C. So yes, Tanner, Anup, you both were correct. The correct answer is option C. Phenotypic and genotypic ratio is similar in case of when the phenotypic and genotypic ratio is similar. Is it complete dominance? Is it incomplete dominance? Is it over dominance, or is it masking, that is epistasis? What is the right answer? when you will find the similar phenotypic and genotypic ratio. It is incomplete dominance, because in incomplete dominance you were getting the same phenotypic as well as the genotypic ratio, and that was 1 is to 2 is to 1, 1 is to 2 is to 1. So the right answer for this question is option B. Now we'll go to mutation parts. So what is mutation? Mutation is random and sudden change in the gene sequences. So when random and sudden changes are happening in the DNA fragments or gene segments, that is what called as mutations. So who is causing this mutation? Mutagens. So can we say mutagens are agents responsible for causing these mutations? Agents or factors. So how many mutagens are there? So these mutagens, they are classified into three categories. One is chemical, other is physical, and the third one you can consider it as biological. So what are biological mutagens? It is virus. So what is coming under physical mutagens? Physical mutagens can be of two types: ionizing or non-ionizing radiations. Or chemical mutagens, it can only cause a mutation in replicating DNA. Now what is this replicating DNA? If this mutagen is affecting the S phase of DNA synthesis, that is what called as replicating DNA, because this is the phase where the DNA replication happens, where the exact copy of the DNA is made. That is what called as replicating DNA. So when the mutagen, they are affecting the S phase of DNA synthesis or DNA replication, this is what called as mutation to replicating DNA. But when it is generally affecting all the stages of cell cycle, whether it is G1, S, G2 and F, then we call it as general mutation to non-replicating as well as replicating DNA. So we'll see it one by one. So firstly, coming to ionizing and non-ionizing radiation. So what is non-ionizing radiation? The good example of non-ionizing radiation, it is UV radiations. So suppose if I'm having a sequence AG, TT, and another DNA sequence like A will always pair with T, G will pair with C, T will pair with A, and again T will pair with A. So how this pairing is taking place? But what happens if the UV radiation is there or DNA is exposed to this UV radiation? So T, that is thymine, instead of pairing with this adenine, it pairs among themselves. So what happens? There happens a pairing between T and T. This T will not pair with A, and again this T will not pair with A. This T will pair together, and this will lead to the formation of thymine dimer. So what is the consequences of the formation of this dimer? If the thymine dimer is forming in the body, it will lead to a skin cancer. The skin cancer will be caused. And what are the other consequences of it? It will also lead to cornea inflammation. So this is what about non-ionizing radiation. So what are the examples of ionizing radiations? So what all examples are there? Gamma rays, alpha rays, beta rays. So why they are called as ionizing radiations? Because once they fall on a matter, they will ionize it. Ions may convert kar deta hai. They will ionize the matter. That is why they are called as ionizing radiation. Why they are doing so? Because they have the higher penetration power. The penetration power of ionizing radiation is always more than that of non-ionizing radiation. That is why it causes the ionization of a matter. So here you need to learn about gamma, alpha, beta rays. So just the examples are being asked in exam. Among this, what is the example of ionizing radiation? So out of this, you have to select even if it is gamma given, alpha given, or beta. And for non-ionizing radiation, what the example is that you have UV radiation. Now let's coming to chemical mutagen. So under physical, we were having ionizing as well as non-ionizing radiation. So non-ionizing radiation, we have UV rays, and ionizing radiation, we have X-ray, alpha, beta, as well as gamma rays. So coming to chemical mutagens. So we have two chemical mutagens here. First is acridine orange, and the other is base analogues. And again, under base analogues, we can have nitrous acid, hydroxylamine, and many more. So how acridine orange is working? Suppose this is DNA. And in DNA, we have major groove and also we have minor groove. So what will happen? Instead, like bases, they are present in these grooves, right? Like T pairs with A, G pairs with C. So what happens? Instead of pairing of these bases, acridine orange comes and it will intercalate and binds to the minor groove of DNA. So there happens no binding between the bases because acridine orange is coming and binding and intercalating to the minor groove of DNA. So this is how acridine orange is working. It will always intercalate to the minor grooves of DNA. Now what are base analogues? Base analogues, we have nitrous acid. So what are base analogues? These are analogues that have structure similar to normal bases. They have structure that are similar to normal bases, but they are not exactly normal. They are mutated form of it. So how this is working? Suppose this is nitrous acid. So normally A pairs with T. But when this adenine, it is exposed to nitrous acid, it will be converted to hypoxanthine. So normally A pairs with thymine. But if it is exposed to nitrous acid, it will be converted to hypoxanthine. Now this hypoxanthine will pairs with C. So you can see a major difference. Previously there was a double bond between A and T, and now you can see a triple bond between hypoxanthine and C. How adenine is converted to hypoxanthine and it is binding with cytosine. What is the another example for this? Suppose this is G. It is having a triple bond with cytosine. What is G here? G is guanine. But when it is exposed to nitrous acid converts this guanine to xanthine. And now this xanthine again pairs with cytosine, but this time the cytosine is different. It will have another chemical adduct associated with it because it is not the normal guanine here. It is xanthine that is pairing with cytosine. Again, it will change the sequence of the DNA. So this is how chemical mutagens are working. So here it can be acridine orange, it can be base analogues too. But acridine orange, it is always for replicating as well as non-replicating DNA. It can go for disturbing the S phase or any phase of cell cycle. But base analogues, they are always for non-replicating phases of cell cycle. That is G1, G2, and M. So if you want to disturb the DNA sequence in the replicating phase, you have to provide acridine orange. But for here, to non-replicating, that is G1S, G1, G2, and M, you have to provide acridine orange, or you can also provide nitrous acid. So now How these mutations are being caused? There are different ways, or mainly three ways how mutations are being caused. Firstly, you can have a mutation in gene sequence. If you are having a mutation in gene sequence, it can be a substitution mutation or a frame shift mutation. You can also have a changes in a chromosomal aberration. Now, if the whole chromosome is changed, what is a chromosome? When the DNA are pegged together, it is forming a chromosome. If the chromosome is changing, so what will happen? It will lead to aberrations, and these aberrations will further lead to cancers. So what are the types under chromosomal aberration? It can be deletion, it can be duplication, it can be translocation, or it can be inversion. Or you may also have a changes in chromosome number. So what are the types under this? One is aneuploidy and other is euploidy. So we'll see this one by one. What is substitution mutation? Substitution mutation, when one, suppose this is ATGG and this is TACC. When one single nucleotide base, it is replaced by any other base. So suppose here G is replaced by T. So what will happen now? This sequence will be changed here. How? Now the code becomes ATTG. It will be changed to TAAC. So you can clearly see how the third base is changed. So the changing of this base is what called as substitution. You can also call this substitution mutation as point mutation. Now under point mutation, we again have two types. It can be transition mutation or it can be transversion mutation. So what is transition? When a purine is replaced by a purine, it is what called as transition mutation, or a pyrimidine is replaced by a pyrimidine. So what are the examples of purine? Purine का examples क्या है? It is A and G. And what are the examples of pyrimidine? It is cytosine, it is thymine, and also uracil in case of RNA. So when purine is replaced by purine or pyrimidine by a pyrimidine, that is what called as transition. Then what is transversion? When purine is replaced by a pyrimidine, or pyrimidine is replaced by another purine. This is what called as transversion. So the last second category we have is frame shift mutation. So what is this frame shift mutation? Suppose in this sequence only, if one base is getting deleted, if one base is getting deleted, it will shift the frame. It will shift the genetic code. Now the final code becomes ATG and here it becomes TAC. So we all know that the protein that is formed is due to the triplet codon. Three bases together, they code for one protein. So suppose if this protein is deleted in between, now the amino acid sequence will be changed. This base sequence will be changed. Amino acid will be formed differently and there will be a different protein in our body. This is what called as frame shift mutation. So if any base is deleted or added, it will lead to a frame shift mutation, because if it is shifting the frame of a genetic code, that is why it is called as frame shift mutation. What is deletion? When some part of a gene is deleted. So suppose this is A, B, C, D, E gene, and suppose from here D and E is getting deleted. Now the frame is again deleted. Now you will have one type of protein because you have deleted D and E from the sequence. This is what called as deletion. And the common example of this deletion is Cry-do-chat syndrome, where there is a deletion in the short arm of chromosome number 5. So what are the symptoms of Cry-do-chat syndrome? The baby will cry like a cat. So this is why it is known as Cry-do-chat syndrome. There happens a deletion in the short arm of chromosome number 5. So here we have two arms of chromosome, two arms that are present in chromosome. One is P and one is Q. P is always standing for peptide, that is a short arm. So whenever there is a deletion in the short arm in chromosome number 5, this will lead to Cry-do-chat syndrome. And if D and E is getting duplicated, this will lead to duplication. And what is translocation? Suppose the common example I'm taking, Suppose this is chromosome number 9 and this is chromosome number 22. There happens a translocation between both these chromosomes. Some part of 9 it is getting exchanged with 22 and some part of chromosome number 22 it is getting exchanged with 9. That is what lead to translocation. So generally the translocation or pairing it is always happening in homologous chromosome, but whenever we are talking about translocation here the crossing over it is taking place between non-homologous chromosome, the chromosome that are not at all similar to each other. Then why here the translocation is happening? Because the chromosome number 9 it bears a telomeric sequences at the terminal, and also chromosome number 22 it bears a telomeric sequences. Now this telomeric sequences are somewhat similar, that is why it is undergoing translocation phenomena. So what will happen if they undergo translocation? They will lead to the formation of a Philadelphia chromosome. Philadelphia chromosome. And this is a worst condition that is happening in the body because it will lead to chronic myelogenous leukemia. It will lead to cancer. How cancer is causing? Because there were telomeric sequences that were similar in both the chromosome number 9 and 22. And isliye there happens a non-homologous recombination. They both were not similar, but look-wise it was similar, and there happens a non-homologous recombination. That is what called as translocation. Then coming to aneuploidy and euploidy. Aneuploidy, if there is a change in chromosomal number, that is what called as aneuploidy. What is aneuploidy? Change in chromosome number. When the chromosome number is changed, that is what called as aneuploidy. Then what is euploidy? When the whole chromosome it is getting extra. It is getting extra or deleted in the body. It is what called as euploidy. Now we'll understand what is aneuploidy here. So suppose we have 2N. All the traits we are having at as a 2N. Matlab AA, BB, they are always diploid. That means 2N. So what is nullisomy? So these are the examples of aneuploidy. Nullisomy, you will have the deletion of 2 chromosome in number. How they are deleted? You can see in the above diagram 1, 2, 3, 4. And here you see it is deleted. Both the chromosomes are deleted. That means the complete diploid set is deleted. That is what called as nullisomy. Null means 0. How 0? Because previously it was 2N. Now 2 chromosomes are deleted from here. That is what called as nullisomy. What is monosomy? Mono stands for 1. And if I'm talking about aneuploidy, we are dealing with chromosome number, not complete chromosomal set. This is a complete chromosomal set, but we are dealing with chromosome number. This is 1 by 1. So what is monosomic? Mono stands for 1. So only you can see 1 chromosome is deleted from here. This is what called as monosomic. That is 2N minus 1. What is double monosomy? Monosomy is happening. 1 is deleted, but from 2 pairs. You can see from here also 1 is deleted, here is 1 deleted. So the formula becomes 2N minus 1 minus 1. What is trisomy? When one of the chromosome set bears an extra chromosome. Tri means 3, but we have already studied normal may, it is always diploid, that is 2N. Now if it is getting trisomy, 1 extra will be there. So it becomes 2N plus 1. What is tetrasomy? Similarly, it will become 2N plus 2, and double tetrasomy 2N plus 2 plus 2 according to the original diagram. Now what is euploidy? When there is a change in whole chromosomal content, it is known as euploidy. We'll see how this is happening. So again, normally standard state we are having diploid, that is 2N. All the chromosomes they are in pairs. You can see they are always in pairs. So if the monoploidy happens, ploidy means change in chromosome set. So if mono means 1, they all will become 1. Now we are not dealing with chromosome number. We are dealing with whole chromosomal set. Jitna bhi chromosome will be present in the body, they will become monoploid. सब एक एक के फॉर्म में हो जाएगा. If polyploidy will happen, if it is triploid, suppose, so what will happen? 2 will become 3. So extra 3 copy it will be gained in all the chromosome set. And if it is tetraploid, that means 4, and this 2 will become 4 everywhere. And here it was happening, it was removing and adding one by one. But here we are dealing with the whole chromosomal content, that is what called as euploidy. And if we are dealing with chromosome number one by one, that is what called as aneuploidy. I hope this concept is clear to everyone. If you have any query, you may ask. Welcome, Srivani. Yes, we'll come to Down syndrome. So the next question you are having, the Cry-Do-Chat syndrome is caused by change in chromosome structure involving deletion, duplication, inversion, or translocation. Why the Cry-Do-Chat syndrome is happening? The Cry-Do-Chat syndrome is happening because of a deletion of a chromosome, duplication of a chromosome. Right. It is because of the deletion of a chromosome, because we have already seen that if short form arm of chromosome 5 is deleted, that is if P part is deleted, it will lead to the cry-do-chat syndrome in babies, where baby will cry like a rat. The sound will appear like a cat is crying. That is why it is coming under deletion theory. Now, chromosome complement with 2n-1, it is called. If the chromosomal complement is 2n-1, what it is called? Is it monosomy? Is it trisomy? It is nullisomy, or it is tetrasomy? If you have the chromosomal content that is 2n-1, what will be it called? Monosomy, trisomy, nullisomy, or tetrasomy? It is monosomy, because we have studied if one chromosome is deleted from 2n and it will become 2n-1. That is what called as monosomic condition. The mechanisms that causes a gene to move from one linkage group to another. When a gene is moving from one linkage to another linkage, it is called as translocation, crossing over, inversion, or duplication. What is the phenomena that is called when one gene is moved from one place to another? It is called as translocation, because we have seen in the previous example where chromosome number 9 and 22, where part of these both chromosomes were exchanged because of the similar telomeric sequences. And this is how the movement of gene is happening. So this is known as translocation. So now coming to disorders. So if I'm talking about genetic disorders, I'm focusing on the disorders that is carried through the gene or through the genotype of the parents. So under genetic disorders, we have two kinds of disorders. One is Mendelian disorders and other is chromosomal disorders. So firstly, we'll focus on what is Mendelian disorders. So firstly, we'll understand this Mendelian disorder one by one. So the very first example in this is color blindness. So what is color blindness? Color blindness is a X-linked recessive trait. X-linked recessive trait. That means this trait is bare on the X chromosome, but it is expressed when it is recessive in character. So it should always be present on both the chromosomes, and then only it will express. So here, if the person is suffering from color blindness, he will be unable to distinguish between red and green color. He will not be able to see the difference between red and green color. But he is always able to distinguish between green and yellow color. This is what known as dichromasia, where the person is able to distinguish only the yellow and the green color. This is known as dichromasia. But sometimes the condition is adverse. The person is only able to distinguish between gray and white color. Matlab everything will appear gray or white. This is what called as monochromatia. Chromatia here stands for disease. Chroma means color. Mono means one. That means the person is able to distinguish only between gray color. That is what called as monochromatia. Now we'll see how this mechanism is happening. Suppose we have a female, that is female ka genotype. What is the genotype for female? It is XX, and if it is bearing color blind ka gene, and suppose female is also bearing a gene for a color blind, so now the male here is hemizygous. Why hemizygous? Because it has one X and one Y chromosome. Hemi stands for half, so hemizygote, the zygote is half. It has one X and one Y chromosome. If the male will bear one C, one colorblind C on the X chromosome, that is what called as infected male, because the male is colorblind. Male is hemizygous, and this colorblind, it is always expressed on a X chromosome. So male is only having one X chromosome. That is what called as hemizygous condition. But what in case of female? So now here the female is having two X chromosome, but if female will have one X chromosome as a colorblind, she will be called as a carrier. But if both the genes, both the X chromosome, if she is carrying this colorblind gene, it is called as a infected or colorblind woman. Okay, so now we'll understand this mechanism, how this inheritance is taking place in case of color blindness. So suppose the female is a carrier here, XXX, or female is a infected male. He is having one colorblind gene on X chromosome. So what will happen? XC, how the gametes will be formed? XC and X, and here again XC and Y. So when we'll draw the Punnett square, what we'll get? We'll get XC-XC condition, XC-X condition, XC-Y condition, XY condition. So here you can see the male that is formed XY. This is a normal and healthy male because it is not carrying any color blind gene. But here you can see XCX. That means the daughter is formed from these parents, and that too she is a carrier because she is carrying only one color blind G on an X chromosome. Only one X chromosome is involved for this. So it is a carrier. But here she is a infected, like she is having color blindness disease, and here again the boy is having, this is XY genotype, but here you can see X bearing color blind ka gene. That is how it is infected boy. So here you can see when the father is infected, because only one X chromosome is there, and that too bearing colorblind G. If the father is infected, the chances of infection in girls is higher. So this is what is called as crisscross inheritance. When the father is infected, there will be a chances to get this color blindness in his daughter. And now when this daughter will become a mother in the next generation and she will have child, that boy will again be color blind. And similar goes with mother. If the mother is infected, it will give this infection to his son and again to his granddaughter. This is how it is going. This inheritance is also known as criss-cross inheritance, from father to daughter to grandson, from mother to son to granddaughter. So always X-linked recessive characters, they always follow criss-cross inheritance. Criss-cross inheritance. You can always see in case of X-linked recessive trait. So can we say that for a girl to be colorblind, अगर एक girl को colorblind बनाना है, father should always be infected, but at least the mother should be a carrier for it. You can see in this diagram also. ठीक है? The father is infected, but the mother is carrier. For the girl to be colorblind, the father should be infected or colorblind, and at least the mother should be a carrier for it. So this is how it is going for color blindness. So I hope this is clear to everyone. So now let's proceed further. Hemophilia. So hemophilia is again a blood related disorder. Blood related disorders. So we'll go fast. Here we have two types of hemophilia, hemophilia A and hemophilia B. If there is a lacking of clotting factor, that is clotting factor 8, so it is called as hemophilia A. It will lead to a disease called as royal disease. And if there is a lack of clotting factor 9, that is Christmas factor in body, that will lead to hemophilia disease, hemophilia B, and this is what called as Christmas disease. So when these clotting factors are absent in a body, this will not be able to clot the blood property, and that's what it is known as hemophilia. Hemophilia, again, it is a X-linked recessive disease. So here you can see normal blood vessel clotting is happening, normal blood vessel, but here hemorrhage, if it is happening hemorrhage, so it will be unable to clot the blood. Why? Because of hemophilia. They are lacking the clotting factor 8 as well as 9. So again happens with sickle cell anemia. What is sickle cell anemia? It is an autosomal X-linked recessive disease. Autosomal recessive disease. So these disease, the link for this disease, it is present on the autosomes, not sex chromosomes. So what happens here? The 6 amino acid, that is a glutamic acid, it is replaced with valine. So this is also called as substitution mutation, point mutation, or missense mutation. Why missense? Because earlier it was forming an acidic amino acid, that is glutamic acid. Now it's converted to aliphatic amino acid, that is valine. So we'll proceed further. We'll go very fast because time is not there. So what is phenylketonuria? Phenylketonuria is a condition. It is again an autosomal recessive disease where it is carried on gene number 12. So what happens here? This phenylketonuria condition, it inhibits the formation of phenylalanine tyrosine hydroxylase. Now this enzyme is absent here. It will not convert this phenylalanine to tyrosine. And if tyrosine is not formed, this will lead to the accumulation of phenylalanine in the body. And at some point of the time, this phenylalanine is converted to phenylpyruvic acid. When this phenylpyruvic acid is converted and it is accumulated in body in larger amount, you may have several symptoms for this. Now, what are the symptoms for this phenylpyruvic acid? You may see a dark color in your urine. So if the phenylpyruvic acid is higher in amount, so the urine color will be dark in color. Now, if it will reaches to brain, that is neural tissue, it may also lead to a condition called as mental retardation. You can also see here in the diagram, at some point of time, this tyrosine is converted to melanin, the pigment that is responsible for hair color as well as skin color. But the enzyme is not there that will lead to the transformation of this phenylalanine to tyrosine. That means tyrosine will not be formed, and if tyrosine is not formed, it will not lead to the formation of melanin. So the patient that are having the phenylpyruvic acid more in concentration or suffering from phenylketonuria disease, they will have a loss of melanin pigment, and there they will suffer a problem of hair loss. Hair color will be light, and as well as they will have the problem of skin pigmentation. So again happens with thalassemia. What happens in thalassemia? It is a quantitative blood disorders. There happens a no presence of platelets in the body. So hemoglobin, we know that it contains alpha 2 and beta 2 chains. So here in thalassemia, this chains, they are getting replaced by beta, and beta is getting replaced by alpha. So when these are happening, this condition is occurred, it will lead to thalassemia. Here occurs anemia. That is what called as thalassemia. So now we'll deal with one or two questions and then we'll end the session. So hemophilia is more common in males because it is a recessive character that is carried by Y chromosome, dominant character by Y chromosome, dominant trait that is carried by X chromosome, or a recessive trait that is carried by X chromosome. So hemophilia is a X-linked recessive disease. So it is a recessive trait that is carried on X chromosome. So the correct answer for this question is option D. So if a color-blind man marries a woman who is homozygous for normal color vision. So what the question is saying, if the color-blind man, so let us draw a diagram for it. If a color-blind man, so suppose this is a color-blind man, if it marries a woman who is homozygous for normal color vision, so the woman is normal here, what is the probability of their son being colorblind? Now what is the question? Sun being colorblind. Only this is our sun being colorblind. So now let us draw a Punnett square for this. So we'll have XCX, this is girl, we'll omit XCY, XCX, and again XY. So what is the probability of sun being colorblind? It is 0, because we can clearly see the boys that are formed, they are normal but not colorblind. They are not carrying any colorblind gene on the X chromosome. That is why the answer for this question is C, that is 0. If a colorblind woman marries a normal vision man, their sons will be. So again, if a colorblind woman, if she is married with a normal vision male, what will be the sons? What is the probability in their sons? So again, XCX will be formed, XCY will be formed, XCX and XCY. We only have to focus on males. So what is the genotype of male? XY, X and Y. And you can clearly see both the sons are affected. So what would be the answer for this? All colorblind sons will be formed. All the sons will be colorblind. So the right answer for this question is option D. So we'll practice one more questions. What will the probability of daughters to be our carriers? So they are asking about this condition. What will be the probability of daughters to be a carrier when a hemophilic carrier man, hemophilic carrier woman, if she is married to a hemophilic man? If she is married to a hemophilic man. So XH, XH, XHY, XHX as well as XY. So we are considering this possibility, hemophilic carrier. So what is the possibility for this? So here we'll omit this because they are sons. Here we have to only answer about daughters. So what is the daughter here? XHX and XHXS. And out of this we are about to answer for probability about carriers. So now these are carrier and they are infected. So what is the probability? So let us consider this as a 100%. So what is the probability? 15% probability for a carrier. So the right answer is option C. So I think we are done with disorders of Mendelian disorders. And this is a table for chromosomal disorder where you have to recall only about Down syndrome, Klinefelter's and Turner. You can just see what are the chromosomal abnormality that are related with this syndrome. So now we have come to an end. So thank you everyone for watching this session. If you like the session, do like, share and subscribe. And the next class, that is on Tuesday, will come up with the series for inorganic chemistry. So till now you have already done a revision of organic part, physical parts. Some topics are already been covered, but inorganic topic is not yet started. So on Tuesday you will be having a revision for inorganic part. Stay tuned and connected for the revision of chemistry. So thank you everyone for your time and patience. If you want to know the 5 previous years NEET analysis, you can just find a link in the description box. Just visit that link. You will find a complete analysis of a NEET exam, and that too for previous 5 years analysis. So for 5 years analysis, if you know, you can just find the description box. So thank you everyone once again. Meet you in the next video. Till then, bye-bye. Take care.